All right. In this particular video, I want to focus more on the structure of the nephron and a little bit about its functionality. And then in another video, I want to talk more about basically with a little more depth, basically just kind of how the nephron works, how renal handling of filtrates and tubular fluid occurs and all that. But this one is more just kind of devoted, again, as I mentioned, the structure and function of the nephron. All right. So the nephron is essentially the functional unit of the kidney. So the functional units of the kidneys. Now, remember that term functional unit, it's been a while since you've heard it, maybe the first time you're watching this video. I just think it's important to understand this term. Remember, the functional unit is the smallest part of an organ that gives function. All right. So the nephron, like any other functional unit, is made up of certain parts that come together that when you put these basic parts together and form one of these nephrons, that basically is what allows the kidneys to do what they do. And these separate components that form the nephron independently or on their own would really not be good, wouldn't really function on their own. So we need to put them together in certain units. Like for example, when we talk about the sarcomere with skeletal muscles, with your Z discs and then your thin filaments and then your big fat thick myosin filaments, these contractile proteins. We need to put them together in a certain arrangement in order for them to actually um, give muscle, like skeletal and cardiac muscle, their functionality, their ability to contract. All right, so just keep that in mind. That's what a functional unit is, the smallest part of an organ or a machine that essentially allows it to function. And remember, the primary function of the kidneys essentially is filtration. Filtration of blood and excretion of metabolic wastes. All right, excretion of metabolic wastes and essentially anything in excess within our bodily fluids. All right. So that's essentially what the kidneys do. So the nephrons are essentially the microscopic structures in the kidneys that we circulate blood towards, and the nephrons are what filter blood and then capture the fluid out of blood, and then essentially through certain molecular processes process that filtrate into which we call tubular fluid once it's actually inside the nephron, and basically removes metabolic waste and excess water, electrolytes, and other materials, and then we excrete all of that material um, via urine out of the body. So the kidneys are excretory organs, and basically they're designed to keep our body fluids clean and free of metabolic waste and any other materials that are in excess that we don't need. All right, and on average, there's about 1 million nephrons per kidney. And we'll talk about the structure of a nephron in a second here. But there's about 1 million of these functional units in each kidney. And a little interesting fact about the nephrons is once we hit about the age, once we get up to about the age of 40 years old, um, we lose about 10% of our nephrons per decade. Okay, we lose about 10% of our nephrons per decade. All right, now what's neat about this, though, it's not neat that your kidneys are degenerating like this, but the nephrons, which I'm not going to talk about much in this video, but in the next video, these functional units of the kidneys are very, very adaptable. So you could be losing nephrons you lose these nephrons, so many per decade. They could degenerate with certain diseases that affect the kidneys, but it takes a lot You have to, I mean, you really don't start seeing the, fairly dramatic effects of losing these functional units until you get down to about, 25 to 30% of your nephrons left inside the kidneys. And that's important because of how adaptable they are. I mean, think about how many people you've ever known or seen that go through life with only one kidney, or maybe start out life with two kidneys and have to remove or have to have one removed for whatever medical purposes. And still live normal lives. So that's one nice thing about the kidneys is they're very adaptable organs, and that adaptability also plays into some of the secondary roles of the kidneys in regulating blood pressure and blood volume and blood essentially regulating blood pressure by regulating blood volume and so on. So the kidneys are very useful organs to the body, and they're organs that we cannot live without. So their adaptability is very important. Um, Now, when we're talking about the nephrons, these functional units of the kidneys, we can break them down into two major components. And those two major components would be the renal corpuscle and then the renal tubules. All right. And both of these are important in both of these parts or kind of sections are important in their own right. So the renal corpuscle, basically what it is a combination of a tuft of capillaries and this structure right here called Bowman's capsule. And the tuft of capillaries, I'll talk about that in a little bit, are called the glomeruli or glomerulus for singular. All right. Now, what you need to know about the renal corpuscle is that this is where blood is filtered. And this would be a good point in time to kind of stop for a second. And if you don't understand renal blood flow yet, if you haven't studied it, if you haven't gotten it yet, you need to go back and understand how blood gets into the kidneys because you'll understand that the circulation of blood is always up into the cortex and for where that's where the blood is essentially filtered, and that's also where all of these renal corpuscles are located, and all these capillaries are. And by now, by the time if you're learning about the urinary system, you understand capillary fluid dynamics and such. But you need to understand how blood flow gets into the kidneys and the functionality of the kidneys. And whether you're a student who's already been going through this, or if you're watching my videos to help you learn, you need to go back and watch my videos about the functions of the urinary system and renal blood flow before you continue any farther into this topic because you're just going to be, you'll kind of be lost or you just won't really truly appreciate the material that we're going to cover. All right, but the renal corpuscle is where blood is going to be filtered, and then the renal tubules are basically where I like to think of as where we process the fluid that we filter out of our blood. Okay, this is where we process the fluid we filter out of our blood because as you remember, as you recall, I've talked about this in a previous video when I talked about the glomeruli, the capillaries that we use to filter blood. They're highly specialized capillaries that remember contain, like any other capillary, endothelial cells, but these endothelial cells have fenestrations or small pores in them, and then a negatively charged basement membrane. And then what makes these capillaries truly unique is the presence of podocytes, or these cells with these feet-like extensions that kind of interlock themselves around the pores of capillaries, okay? And the purpose of this design is to allow only very small materials, basically water and very small materials, to be filtered out of our blood. That's why people really don't have any protein or cells in the urine. They should have no cells in the urine, and there might be just a smidgen of protein in people's urine, like less than a percent of the total composition of materials in there would be protein, or less than half a percent. But so you have to keep in mind that anything that's small enough to be filtered out of the glomeruli will be filtered into the nephron. All right. And that means not just the metabolic waste products, but also products that are useful to us that we want to retain in our bodies. All right. And that's why it's important that we have these renal tubules to, when I say process that fluid, basically reabsorb that material back into the bloodstream. And there are wastes that are going to be secreted into those tubules as well, because we don't necessarily filter out all of the waste immediately in at the renal corpuscle. So on that note, let's kind of take a look at the nephron itself. So what I'm trying to do here is, so here's a kidney. This was a pig kidney that we dissected in our lab, And you remember your basic, your two major areas of the kidney. You've got your renal medulla, which consists of the pyramids, and then the renal cortex, which is basically all the tissue surrounding the renal pyramids, i.e. renal medulla. And and basically all of these microscopic tubules, these nephrons, are located within this tissue that you obviously can't see with the naked eye. And we've gained an understanding of these nephrons, obviously, through countless histology studies and analyzing, kind of the makeup of how these, these renal tubules and nephrons are designed. Now, first off, what I want to say is that, so let's focus on the renal corpuscle first. All right. So what I'm going to do is I'm going to use the black marker for this. So first off, I'm going to draw this line right here. Okay. And down, we're going to say that anything below this line would be the renal medulla, or a pyramid, or the renal pyramids. All right. And then anything above this line is going to be the renal cortex. Okay, and that's why I want this kidney sitting right next to you while I'm talking about this material. So, because I want you to be able to picture these parts that I'm talking about, on the kidney itself, right next to you. So remember, as you know, all of this would be the renal cortex, and then these pyramids would essentially be the renal medulla. All right. So first off, the renal corpuscle. Um, the renal corpuscle is going to be located in the renal cortex. All right. And the renal corpuscle consists of two parts. And I apologize ahead of time for my art, but unfortunately you're subjected to it if you're watching my videos. So the renal corpuscle, as I mentioned, consists of two parts. All right. This structure that's called Bowman's capsule. All right. And then the glomeruli, these capillaries that are located within, encased within this capsule. All right. And remember, entering the renal corpuscle is going to be the afferent arteriole, and then exiting will be the efferent arteriole. Now, I know, again, this is kind of horribly drawn, but what you have to bear in mind is that the afferent arteriole is always going to be larger in diameter than the efferent arteriole, and that plays a very important role in the regulation of renal blood flow and how much blood is actually flowing into these capillaries, and how we're going to be filtering, and how much blood we're essentially going to be filtering. Okay, so so basically, as we circulate blood up through the kidneys, it is going to be, it's going to pass through the afferent arteriole into these glomeruli. All right, and then the fluid will then be filtered out of these capillaries and then into this. Notice how there's a little bit of space here. I know, again, it's not, the best drawing I've ever done, but excuse me, there's this little bit of space here that's called the capsular space. Now that's important because there has to be room for this fluid to actually pass out of the blood and collect in. And then, now you're probably thinking, well, why do we want this capsule around these capillaries? I mean, when you think of every other tissue in the body, basically there's just, arteries. Should probably draw that in red. Arteries coming in. Um, capillary, that just go right into capillary beds, right into tissue spaces. We filter fluid freely out into the cells of the interstitial fluid of the tissues, and the blood flows right back up through the venous system. The importance of these Bowman's capsules, or what some people call the renal capsules, is that these act as traps. All right, so these trap this fluid, in here, the material that's coming out of the capillaries within the renal corpuscle. That's what we call filtrate. All right, that's what we call filtrate. And then this capsule will trap that filtrate and really only give it one direction to flow. Instead of dispersing all throughout the interstitial fluid of the renal cortex and the kidneys, that will force the fluid, then to actually start to travel into the second component of the nephron, the renal tubules. All right, and then the first renal tubule that we'll focus on is called the proximal convoluted tubule, or otherwise we'll just abbreviate this PCT. All right, PCT. So this would be the proximal convoluted tubule. All right, and then the proximal convoluted tubule is usually gonna, well, not usually, they're gonna have these short little necks, and then basically these kind of long twisted tubes. Now and you have to bear in mind the word convoluted means bent or twisted or whatnot. And we call this the proximal convoluted tubule because, well, this is essentially where where the renal tubules begin. The tubules begin at the beginning, the origin, the renal corpuscle. All right. Remember, proximal essentially is a word. Proximal is a word to describe, points of attachment closer to the midline or closer to a point of origin or whatnot. All right. So the proximal convoluted tubule, and then what the proximal convoluted tubule is going to do is it's going to descend down towards the medulla. All right. And then what you're going to see is it's going to dip down into the renal medulla, and then it's going to form this little loop-like structure here. And then it's going to come back up again. Okay, so basically what we've got down here now is we've got this structure. This would be called the thick descending. This would be called the thick ascending limb. So thick descending, thick ascending. And then we would normally have a thin ascending. I'm just going to abbreviate this, and a thin descending. All right, and then it's going to form this loop where these descending limbs are going to loop back up towards the cortex, and this area where the loop occurs, this is called the loop of Henle. Okay, the loop of Henle. All right, and then what you're going to see is it's going to come back up, and then you're going to form another convoluted tubule, except this one is going to be called the distal convoluted tubule. The distal convoluted tubule because it's another series of bent tubules in the renal cortex, but they are further away from the point of origin, the renal corpuscle. All right, and then the last part, which isn't necessarily a part of the nephron per se, but the last, kind of critical part of this is going to be an area where where distal convoluted tubules are going to converge, or I should say collect. All right, where these distal convoluted tubules are going to collect. And these areas are called collecting ducts. I'm just going to abbreviate this CD. All right, collecting ducts. Now, collecting ducts could have upwards of maybe only 5 distal convoluted tubules draining into them, or there could be well over 1,000 distal convoluted tubules meeting up with the collecting ducts. And then what the collecting ducts are going to do is they're going to dip down in the renal medulla, all the way down towards this area of the renal pyramid, as you remember is the renal papilla. All right, and then fluid will then drain from from all these collecting ducts that are kind of aggregating down at the renal papilla into a minor calyx, and then drain into a major calyx, and then meet up at the renal pelvis, and then out the ureter. And by the time this fluid gets into the collecting duct and is on its way out, now we have urine. Okay, as the tubular fluid, well, as the fluid is passing through the nephron, through all these different parts, and it's being processed, this is tubular fluid. So basically what we're essentially doing with the nephron here is we're filtering blood, and as I mentioned, we're removing water and all those other waste and waste products and other useful materials out as well. And then what we're doing is we are processing or handling that material that's in the tubular fluid. All right, so by the time we're done processing that fluid and it gets into the collecting duct, we basically now have urine. All right, and then that urine is going to drain out of the kidney. That's essentially, remember what these calyces are for. and these are the drainage channels to get this urine out towards the urinary bladder where we will store it for further use. All right. And another thing I wanted to mention is, the inner lining of these tubules is going to be different as you go as well. The proximal convoluted tubule is going to contain, um, is essentially going to contain a simple cuboidal cells, or they look like simple cuboidal cells. They're a little kind of goofy looking, but essentially they are simple cuboidal cells. Now, the simple cuboidal cells that line the proximal convoluted tubule are also loaded with microvilli. Okay, so there's this brush border all along the lining of the inside of the proximal convoluted tubule. Okay, now that's important for you to understand because remember, wherever there are microvilli, okay, wherever there's that brush border being formed by microvilli, that should automatically tell you that this is an area where there's going to be a lot of absorption and secretion of materials. A lot of absorption and secretion of materials, or just in general, at least absorption. All right, because remember, microvilli are cellular structures that increase the surface area of cells. Because remember, in order for absorption to take place, materials have to come in contact with the cells, and then they will be pumped into the cells where we'll do what we need to do with the material that comes in. So microvilli just make the cell a little bit bigger by extending out farther. So these microvilli will extend further into the lumen than just the cells could themselves, and enhance the proximal convoluted tubule's ability to basically absorb materials out of that tubular fluid. So essentially, so what we have going on here, remember I said that we filter out a lot of useful substances out of the bloodstream. This is where the bulk of the useful substances are going to be reabsorbed back into the bloodstream. All right, that's essentially what we're doing. That's the primary purpose of the proximal convoluted tubule, taking in all that useful material that we want to retain. And also, you have to bear in mind that there are going to be peritubular capillaries around here as well. All right, so remember, as blood flows out through the efferent arteriole, it's going to come out and form these peritubular capillaries that are found either, and these capillaries are either going to be found around the proximal convoluted tubule or around the loop of Henle in the renal medulla, and that depends on what type of nephron it is we're talking about, because there are two types of nephrons that can be located within the kidneys. One of those types of nephrons would be a cortical nephron, and cortical nephrons make up about, let's go with 80% of all of the nephrons within the kidney. Now, when I say cortical nephrons, what I mean is that the bulk of these structures are located up very high up in the renal cortex, all right? And cortical nephrons have all of the same structures that we're talking about, but one thing that makes them unique is they have very long proximal convoluted tubules. Very, very long proximal convoluted tubules. Okay? And surrounding these proximal convoluted tubules are going to be peritubular capillaries. Okay? So the bulk of the nephrons in the kidneys, the bulk of the proximal convoluted tubules in the kidneys, as you can see, are devoted to reabsorbing the useful material that we filtered out of our blood that we want to keep. All right? And then the other type of nephron that's located within the kidney would be called a juxtamedullary. Okay, remember juxta means nearby or close to, and medullary, so close to the medulla, renal medulla here. So juxtamedullary nephrons, what's different about these is these have very short proximal convoluted tubules and very long loops, very long nephron loops of Henle. All right. And instead of forming peritubular capillaries, these, the capillaries that surround these, we call the vasa recta. Okay, the vasa recta. All right. Now, basically these nephron loops and these loops of Henle, what these are essentially devoted for is, this is where the bulk of the natural water retention occurs. All right, because even when it comes to water, all right, we don't want to expel all of the fluids that we filter out of our bloodstream as well. So by design, just by design, this is where the water retention occurs. Now, so typically on the descending limb, um, the descending limb is highly permeable to water, and then the ascending limbs are highly permeable to salts. All right, highly permeable to salts. All right, so and I'm going to talk about this with some more depth later on, but essentially what happens here is in the renal medulla, there's a very, very high electrolyte, salt composition within the interstitial spaces because we're constantly leaking salts out of the nephron loop into the tissue spaces. All right, and then as a result, by having all of those salts out here, this is how we maintain it by having one side of this loop very permeable to salt. So many of them will leak out. All right, and then while fluid is traveling down, um, it's going to be, and I said this is primarily permeable to water, but not salt. All the high electrolyte content of the renal medulla is just going to naturally draw a lot of water out relatively easily. All right. So that's essentially, and I'll talk about that with some more depth later on, and that's a process called a countercurrent multiplier. But that essentially is what the nephron loop and the loop of Henle are devoted to. Naturally retaining water. All right. So then what's going to happen is the tubular fluid is, and then again, that's the other major type of nephron that's located in the kidney. And so you can do your math. The remaining 20% of nephrons um, are these what you call them, juxtamedullary nephrons. And as I mentioned, these have very, very long loops of Henle. They dip very deep down into the renal medulla and very short proximal convoluted tubules. Whereas with cortical nephrons, these have very short loops of, nephron loops of Henle, and sometimes the loops may not even get into the pyramids. But remember, they have very big, long proximal convoluted tubules located within the cortex. All right, so those are the major differences between your two types of nephrons. And then what we essentially have here is then that tubular fluid will pass up into the distal convoluted tubule, which is essentially lined with more, cuboidal type cells. Um, just like the loop of Henle as well. Lots of cuboidal cells. Because remember, wherever there's cuboidal cells, there's going to be, I mentioned this when I talked about histology. Wherever there's cuboidal cells, there's going to be a lot of absorption or secretion. Especially secretion more than likely taking place. Now by the time the tubular fluid has worked its way to the distal convoluted tubule, it's been pretty well processed. So, but there will be a little bit of material, some waste here or there, some electrolytes, some acid-base balance occurring here, but very, very minuscule amounts occurring here. There is something important about the distal convoluted tubule you need to understand though, is that the distal convoluted tubules are always going to pass by the glomeruli and the arterioles of the heck happened here? And the arter and the art shoot and the arterioles. Okay. Now that's very important because that forms a structure which I'm going to talk about a little more in a second called the juxtaglomerular apparatus. And there are these specialized cells, these kind of long columnar cells located within the juxtaglomerular apparatus that are called the macula densa. Okay. The macula densa. All right. Now these are important because I'm not going to talk about this in this video, but in another one, I'm going to discuss essentially how the kidneys, remember I mentioned earlier that the kidneys are highly adaptable organs. This is part of their adaptability, is that what the macula densa does, and this is a process that still is not terribly well understood to this day, um, but what we do know is that these cells that make up the macula densa, this little area of distal convoluted tubule, what they essentially do is they They kind of monitor the osmolarity or the electrolyte or the overall, that's what's not well understood, but they essentially measure the composition of the tubular fluid of, when they believe it's either, a lot of people like to think that it's sodium that is monitored, within, monitored by these cells. And basically this is, and these, what that tells us is that is filtration rate too high or too low, because if we filter blood too fast or too slow, within the renal corpuscle, that's going to affect essentially how we process or handle the materials that are in the nephron. And then, depending on what's going on with the tubular fluid, then that will essentially give us an indication of, the right physiologic responses necessary to compensate for the alterations in renal blood flow. All right, and then and then also there are some receptors for aldosterone located here. And as you remember, a lot of, textbooks like to call aldosterone the salt retaining hormone. I'd like to go against that and be a little more specific and call this the sodium retaining hormone. Okay, the sodium retaining hormone, because remember, aldosterone stimulates sodium potassium pumps, within the kidneys, especially within the distal convoluted tubule. And basically what we're doing is we're pumping sodium, out of the tubular fluid and back into the, which will eventually be reabsorbed back into the bloodstream, and potassium into the nephron, where we wind up urinating or peeing out more potassium. All right, but there are certain scenarios when that is important, and I'll talk about those later. Okay. And then the distal convoluted tubule, what's important to know for now about the distal convoluted tubule, this is where you primarily find the receptors for antidiuretic hormone, or other, oops, probably make that look like an H, or ADH. Okay, that is where the receptors for ADH are located. Okay. So know your parts of the nephron. And like I said, it's important that you have this basic information right now and know the difference, and are able to know the difference between a cortical and juxtamedullary nephron, because they are different. I mean, they are made up of the same parts. It's just that these parts are a little different, from nephron to nephron. But it's important that you understand the basics about this for now. All right. Oh, and another thing I forgot to mention, gosh dang it. I forgot to mention that essentially why is my red marker not working? The distal convoluted tubule is lined with cuboidal cells, just like, kind of simple cuboidal cells, just like the proximal convoluted tubule. But remember I said that there's not a whole lot of reabsorption and secretion of materials going on here. These cells, these cuboidal cells, do not contain microvilli. Okay, there's no brush border here on these cells, and that right there should be an indicator that tells you that there isn't a lot of absorption and secretion going on. And then same thing with the collecting duct, just, simple cuboidal cells without a brush border. So basically, you have to keep that in mind. All right. So hopefully that was clear and concise, or kind of to the point enough. If not, definitely ask questions. All right. Now, what I want to talk about next are essentially is just real briefly is the basics of how we handle materials inside the kidneys. All right. So basically, this picture is going to represent the entirety of the two parts of the kidneys. So picture this part, this first part as the renal corpuscle, and then this part down here as all of the renal tubules put together. All right. Now, when it comes to, there are basically four different ways we can handle or process the filtrate that becomes tubular fluid that winds up in the nephron. All right, and you also have to think that there are going to be peritubular capillaries or vasa recta of some sort. All right, along these renal tubules. So first off, what we can do is there can be materials that are filtered out into the renal tubules, and there essentially could be no reabsorption whatsoever. No reabsorption of materials at all. There are some materials that are just filtered right out of our bloodstream, and that work their way through the tubular fluids, and none of it works its way back in into the bloodstream. Okay. Another method that we see is we could have filtration occur and partial reabsorption. Okay, partial reabsorption, which you'll see with, some sodium, some of your electrolytes, some waste products. You will reabsorb some waste, metabolic waste, back into the bloodstream. All right, but that's another form of handling. Okay, another one would be material comes in and total reabsorption occurs. So, all right, and glucose is a good example of this. So glucose, as long as your blood sugar is below about 220 milligrams per deciliter, which is a relatively high number, but as long as your blood sugar stays below that number, you will essentially reabsorb all the glucose that has been filtered out of your blood back into the blood itself. Same thing with amino acids and vitamins and others, kind of these nutrients that are useful to us. Okay. Um, So then now let me ask you this for a second. What will that tell you then about the composition of glucose in the urine? How much glucose will be in your urine if you reabsorb 100% of it back into your blood? Well, that should be an easy answer. None. So normally there should be no glucose in your urine. You're only really going to see glucose in people's urine, like for example, people who are diabetics who aren't taking their insulin or have very high blood sugar and aren't regulating it properly. All right, so you have to keep that in mind. And then the other form of handling that can occur is where there's a lot of secretion taking place. Okay, lots of secretion taking place from the from these vessels into the nephron itself. All right, and you're going to see this with a good handful of metabolic wastes. Okay, with metabolic waste. All right, there are some wastes. I mean, some's going to get filtered out, so we're going to have a hard time with that. But essentially, that's when you'll see this occurring. All right, so bear in mind that these are the four. I know this. I know I just kept this simple and basic, but there just isn't a whole lot of explanation that really needs to go into this. But these are the four ways that we handle or process tubular fluid. All right, again, some materials are 100% retained. Some are partially reabsorbed back into the bloodstream. Some materials are 100% reabsorbed back into the bloodstream, and some materials are purely secreted from the bloodstream into, um, from those paratubular capillaries into the nephron. All right, so you have to keep that in mind. And then what I want to talk, then the last topic that I want to discuss is the renal corpuscle. All right, so the renal corpuscle essentially is basically comprised of this first major part, which is, I'm sorry, renal corpuscle, I'm losing my mind. I want to talk about the juxtaglomerular apparatus. Okay, which is the renal corpuscle plus the distal convoluted tubule. Sorry about that, folks. So the renal corpuscle, remember, is going to be this capsule here, Bowman's capsule, which is, just lined with a, simple layer of epithelial cells, squamous cells, I believe to be specific, simple squamous. Um, and then remember all these number 10s here, these are representing the glomeruli, or these capillaries that are located in here. All right. And then number this right here would be the afferent arterial, and then this would be the efferent arterial, and the arrows alone should indicate that, because remember, afferent means moving towards an object, and efferent means moving away. All right. And then here, what we see, this would be the distal convoluted tubule, and then these particular cells right here would be the macula densa. All right, the macula densa. Now, why it's important to just have a basic understanding of this is, one, because by design, notice how there's this, you've already seen me talk about this a little bit before. Sorry, my red marker's going nuts on me. You've seen me talk about this before. Remember, I mentioned that there's going to be a little bit of space in between the capillaries and the Bowman's capsule itself. Now, again, this space allows for fluid to collect and then be, and then be pushed into the proximal convoluted tubule, where it will then be handled, throughout the, throughout the lining of the nephron. All right. And also the fluid that builds up inside this space also is going to build up a little bit of water pressure, what we call capsular pressure, which I'll talk about more at the beginning of the next video when I talk about glomerular filtration rates. Okay. Now, what's interesting about these cells that are found within this juxtaglomerular apparatus, all right, is, the importance of these is that these help the kidneys auto-regulate blood flow. Remember, auto-regulate. In medical terminology, auto means self. Okay, so the kidneys are very good at self-regulating. All right, and they're very responsive to basically blood flow and blood pressure changes as blood is flowing in through these arterioles, and then the combination of just the blood pressure and blood flow alone, and then you combine that with how the tubular fluid is handled, and then we create this really neat essentially mechanism for the kidneys to regulate themselves. All right. So remember, the juxtaglomerular apparatus is relatively simple. It is basically the renal corpuscle plus this area of the distal convoluted tubule where we've got the macula densa and then some other cells, which I'll talk about more in depth a little later. And as I always say, if you guys ever have any questions, do not hesitate to ask. And, hopefully this video was clear enough for you guys, because, this is just a very, very important, this is just an important topic for you to understand because the kidneys are, I mean, we need them, because the kidneys are, these are the main organs we use to get rid of metabolic waste. These are the main organs that help us regulate body fluid levels. These are the main organs that help us, that they're not the main organs, but these are the strongest regulators of pH. All right, because of their ability to directly excrete hydrogen ions in the urine. All right, and understanding the structure of the nephron and then these basic concepts of renal handling. And then, I'm going to, in the next video, I want to talk about glomerular filtration rates and how that'll affect the amount of fluid that will enter into the nephron and be handled is very important. All right. So if you have any questions, as I always say, don't hesitate to ask. And I guess I'll end this video with a classic anatomy saying, you can eat kidneys, but you have to boil the piss out of them first. Think about that.